Now, the next step is TCR recognition. So, till now, the criminal is arrested by the guards. And we know, here the criminal is the antigen. And the guards are the surface MHC molecules. Now, these guards containing the part of a captured antigen are standing over the surface of the infected cells, or APCs, where they are waiting to be seen by the vigilant eyes of our immune system. And those eyes are actually T lymphocytes. Although these T lymphocytes are the eyes for the adaptive immune system, but they themselves need an eye to detect the antigen MHC complex. And that vision is provided to them by the special surface molecules that are called T-cell receptors or TCRs. These TCRs are actually cell surface proteins found over the surface of T cells that recognize specific antigens, initiating immune responses, and defending against pathogens. 95% of these TCRs are alpha-beta TCRs, made of alpha and beta chains. And 5% of them are gamma-delta TCRs, made of gamma and delta protein chains. Now among these, gamma-delta TCRs can bind the antigen directly. But here, we will talk about majority that are alpha-beta TCRs. So, we say they don't bind the antigen directly, but need an intermediate indicator presenting a part of antigen. That is actually MHC antigen complex over the surface of other cells. So, what happens is that MHC acts as an indicator. In infected or tumorous cells, the cellular-derived antigen proteins or fragments of endogenous antigens are processed inside the cell and then presented over MHC class I molecules. Then, even at non-infected cells, MHC-1 molecules are present. And here, they display self-peptides on the cell surface. And on the surface of APCs, the foreign-derived antigens are processed, and some of its fragments are presented on MHC class II molecules. Now, these indicators holding their particular flags are standing over the surface of cells. Here, T cells that are wandering around will encounter these flags now. So, what will happen? At non-infected cells, where there are only self-peptides attached over MHC, the immune cells, particularly cytotoxic T cells, continue to survey and ensure that the cells are functioning properly, as there is no sign of danger. But on infected cells, where there is a red flag, as some altered or infected proteins are there, the cytotoxic cells will immediately recognize them as danger and will take action against them. Then, in third case, where there are fragments of foreign antigens, helper T cells will come into action, recognize them, detect some danger, and help regulate other cells to take action against them. And this recognition is mainly done by TCRs, where antigen MHC complex binds over the antigen binding site of these TCRs. The TCR recognizes both the peptide antigen and the MHC molecule simultaneously. As the TCR's variable regions make contacts with both the peptide and the MHC molecule, ensuring specificity for both the antigenic peptide sequence and the MHC molecule. And if they are compatible with each other, then they proceed with necessary actions. But here is a twist. TCRs alone are not enough to stimulate antigen binding. So, they require some additional molecules for co-stimulation. That is the next step in T-cell activation. Ever felt overwhelmed by the traditional way of learning medicine? Enter the future of education with digital learning. Whether you're refining your knowledge of biochemistry or immersing in cell biology, it's all there. Understanding complex medical subjects has never been easier or more efficient with engaging, interactive content. Welcome to the new age of medical education. Learn anywhere, anytime, at your pace. Powering your medical journey. sqadia.com